All right, so this is lecture 3, Fundamentals of ID Epidemiology, part 3. And so what I'm going to give you today is more vocabulary. So it's going to be a lot of vocabulary words. Some of these are going to be in Giseki, but there are words that we're going to refer back to over the course of the course. So the first I want to talk about is direct and indirect transmission. There are two types of contact transmission, direct and indirect. Direct transmission is when there is physical contact between an infected person and a susceptible person. So for example, like in this little picture, there are two people shaking hands and one of them has transmitted something to the other person. And examples of pathogens with direct transmission are HIV, which requires sexual contact or direct contact with blood products. Ebola, which again requires contact with bodily fluids on a person. Herpes viruses require physical contact. There's a number of them. And this is really the easiest one to get your head around, direct contact. One person touches another person, either just in shaking hands or hugs or whatever, or sexually, and another person becomes infected. Indirect transmission is when there is no direct human contact. There can be something in between, like for example, I cough and aerosolized particles like drop onto a table, and then you touch the table or the doorknob or whatever, and you pick them up and become infected. So for example, here in the picture here is like a person touching a light switch and a doorknob. Doorknobs are great places to transmit things like influenza. So if you want to get influenza, definitely touch more doorknobs. One great public health intervention might be to have automatic doors. In Japan, they're very serious about this, and have like automatic doors where you actually don't touch anything. I don't know how much impact that has on things like influenza transmission, but that's one way. Droplets in the air, people cough, floating around the air, and then you pick them up, through breathing. Airborne transmission, fully airborne transmission would be things like influenza and tuberculosis, where you breathe and particles go floating in the air and directly infect people. The fecal oral route through food or water, cholera, norovirus, Shigella, these things, where food is contaminated with fecal matter, and you pass on to other people. And then there's the more complicated case where transmission occurs through vectors, through animal vectors, and things like malaria, which are transmitted by mosquitoes, fleas, which transmit plague, a number of vector-borne diseases out there. So direct, indirect. And we're going to keep coming back to this. Here's the picture a little bit bigger from Harvard. I think this is a very nice way to describe how this works. Now, we classify these routes of transmission with direct. I mean, there's like skin-skin contact, which leads to infections of herpes type 1, mucus-mucus, which transmits sexually transmitted infections like syphilis and gonorrhea, transplacental, so we can have what we call vertical transmission, which transmits from mother to child. Through the placenta, via breast milk transmission of HIV, for example. Indirect transmission, examples of this include foodborne diseases like salmonella, waterborne diseases like hepatitis A, vector-borne diseases like malaria, or airborne diseases like chickenpox. An epidemiologically relevant exposure, therefore, depends on the agent. And agents may have multiple routes. There's a number of diseases that have multiple routes of transmission. So, for example, you might have potential mucus-mucus transmission and also airborne transmission. So, for example, like brucella and Q fever are both sexually transmitted, and they can also be transmitted by airborne. They're also transmitted through saliva in contact with bodily fluids, but they can also be transmitted through milk or even water. So pathogens, some pathogens have very specific transmission routes. Others are very flexible. So microbiologists classify pathogens into distinct groups. So I'm going to talk a little bit about what these groups are. The first one you already know about is bacteria. It's one of a large group of unicellular microorganisms which have cell walls, but lack organelles and an organized nucleus. Some can cause disease. Bacteria are important in that they are organisms that require sustenance, and they require sustenance to replicate. So they're really no different than organisms like ourselves. They're just like on a cellular level less complex. Not all bacteria cause disease. There are many bacteria that transmit, like that we need, that we rely on to exist. If bacteria were eliminated from the earth, all life would cease to exist. Your gut is filled with bacteria that assist in digestion. They help do things like protect you from disease, all kinds of things. You have them all over your skin, like in every part of your body. When we talk about pathogens, we're talking about the ones that cause disease. And as you can see in the picture, they come in all sorts of shapes and flavors. Viruses. Viruses are really cool. They are a nucleic acid molecule in a protein coat. And they are interesting because they can multiply only within the living cells of a host. They cannot replicate on their own. They have to take care of the machinery of the cell of a living host to create copies of themselves. There's a debate, of course, of whether viruses actually constitute an organism, a living thing or not. I don't really have skin in the game on this debate, but they are quite fascinating in that they're not like typical organisms. They're really just sort of machines that are designed to make copies of themselves. Examples of viral diseases include HIV, influenza, Ebola, and they also come in all different shapes and sizes. And they don't just infect humans, they infect every single type of organism on the planet. They even infect bacteria. Fungal diseases are really cool. Examples of fungal diseases include athlete's foot and ringworm. There are fungal diseases of the lungs that become very, very serious and very difficult to treat. They're fungus, just like, fungus you see like, on your wall or forest or whatever. They populate, use cells as roots and create infection like this. This is a ringworm infection here. And anyone who's had ringworm knows that it's incredibly difficult to treat. It takes a long time to treat. Athlete's foot is the same. It's not easy to treat. And toenail fungus is another one that's very difficult to treat, and it tends to stick around for a long time. Parasitic diseases are caused by protozoa, helminths, trematodes, etc. These are standalone organisms that have, in most cases, have sexual reproduction. They require sustenance. They require sustenance to reproduce. They often require iron to reproduce. They're organisms just like us in every sense of the word. Examples of this include filariasis, intestinal worms, malaria. Yeah, there are many of these out there, and we're going to talk a lot about these over the course of the term. Prions. are like the coolest, scariest type of, I don't even know, you can't really call them organisms because they're just misfolded proteins with the ability to transmit their misfolded shape onto normal variants of the same protein. Prions are what cause mad cow disease and Creutzfeldt-Jakob syndrome, which unfortunately is spelled incorrectly here. There we go. And these are degenerative brain conditions. Basically, the proteins in the brain start misfolding and basically turns your brain into a soup, more or less. Very fascinating. Prions, you can't autoclave it. You know, you can't, if you do any sort of surgery on a person who's infected with prions, everything has to be disposed of because they're not alive. They can't be killed in the traditional sense. Very fascinating. Very rare. Prion diseases are very rare, but definitely don't get them. They're very serious. Yeah, so here's a picture of a brain infected with Creutzfeldt-Jakob syndrome. So these are sort of the 5 classifications that microbiologists use. Clinically, these are very important because they inform treatment strategies. In terms of ID epidemiology, these classifications are less important to us because we view them all through the same lens. Because they're all infectious, they can be transmitted between humans, and all have their own, even within these categories. I mean, different bacteria behave in very, very different ways. You know, viruses behave in very different ways. You know, for example, like, you know, Ebola is vastly different from influenza. COVID-19, you know, is vastly different from, you know, many other viruses. Fungus, there's many different types of fungus. Parasites are all over the place in terms of behavior, being independent organisms on their own, you know, sort of, you know, their own distinct evolutionary trajectories, and prions are just weird. So you're going to be overwhelmed by a lot of new terminology that you hear over the course of this course. If you have any questions on terms that I use or you see in the PowerPoints, you know, please speak up. You know, things like attack rate, immunity, vector, transmission pathways, carrier states, subclinical disease, repeat episodes, resistance, clone, immunity, serotypes, strains. These are all things that we're going to go over through the term. So reservoirs are basically the place from which disease arises. It has to sit somewhere to be accessible by humans. That's when we talk about a reservoir, that's what we mean. Human reservoirs, obviously right now, COVID-19, you know, we're reservoirs for COVID-19. You know, any sort of, you know, these classic sort of infectious diseases that you think about, you know, like smallpox, HIV, influenza, COVID-19, gonorrhea, these different things have human reservoirs. The next is zoonoses. These are diseases, pathogens that are transmitted from animals to humans. These are quite fascinating because there's all different sort of complications that are presented by having an animal reservoir like this. Examples would be rabies, Epstein-Barr, brucella, tularemia. There's many. West Nile. There's many. Yeah, there's many. And they're all different for sure. And zoonoses are right now, I think, you know, the most important diseases, pathogens out there. HIV was originally a zoonosis. It jumped from animals to humans. These things like rabies, for example, don't transmit well from human to human because we tend to isolate ourselves. You know, we tend to isolate people with rabies. You know, brucella, human transmission of brucella does occur, but it's rare. Yeah, so human reservoirs, zoonotic reservoirs. Soil can be a reservoir. So for example, histoplasma, clostridium, tetanus is a soil is it soil-based pathogen. Anything you can get from the soil, water, reservoirs. There's a number of waterborne diseases out there, cholera, for example, Legionella. These classifications can be slippery and uncertain. The epidemiologic impact of environmental cholera reservoirs is not well defined. And guinea worm appeared to not be a zoonosis until very recently. We're going to talk about guinea worm later. I think in ID epidemiology, you know, with those microbiotic classifications of diseases and these classifications of reservoirs, you really have to take this with somewhat of a grain of salt. They're intended to be a guide, but they're constantly changing. ID is slippery that way. It changes a lot. And what was, you know, a zoonosis at one point can become, you know, a human transmitted pathogen in the future. So we have to be careful. So please don't look at these in isolation from one another. The distinction between these are much grayer than what this would suggest. So again, we're talking about this epidemiological triad of host, agent, or pathogen in the environment, and how these things come together to create conditions of disease. And this is a picture we're going to keep going back to over the course of the term. Going into more depth on the triad, you know, with agent, the pathogen, you know, we have infectivity, pathogenicity, virulence, which is how serious the disease can be, immunogenicity, how likely is it to produce an immune response in a host, antigenic stability, survival, how long does it last? The host, we have age and sex. We have the particular genotype of the host. What is the genetic profile of the host? I think this is very important. The behavior of the host makes a huge difference. Nutritional status of the host. I mean, people who are malnourished are going to be more susceptible to disease than other people who are well nourished. And general health status of a host. And environment can be things, you know, as simple as weather. It can also be the type of housing you live in. The geography, what's the terrain look like? Is the terrain sort of amenable to, for example, mosquito reproduction? Or, you know, amenable to polluted water? Occupational setting, are people working? Is their work itself leading to increased opportunities for transmission? Air quality, we find that, you know, lung infections tend to be higher when in areas of heavy air pollution. Political unrest, warfare, conflict, all these things can create conditions for disease, as well as, you know, especially what we're seeing here in the United States is political unrest and political discord is hindering the ability to create policy that could protect the public health. I mean, we've seen, you know, very little serious public health policy in the past 4 years. It's actually quite disturbing, if you ask me. Infrastructure, how well, you know, is the built environment work to prevent, or, you know, how well does it work to exacerbate disease? All these things. So host, agent, environment triad is going to be very important to understanding about infectious disease in this course. The triad is dynamic, and in what we call endemic scenarios, it can be balanced. We're going to talk about endemic, epidemic, and pandemic later. Sort of, you know, right now, just a easy definition of it is when you have an outbreak of something, it can become epidemic. So it's an outbreak. Endemic means that there's sustained transmission within a community. And then pandemic means that it's everywhere. But, in an endemic scenario, often humans and pathogens can live in some sort of balance with one another. But this is not going to be the case for new pathogens. Like, for example, with COVID-19, we hope, that we can, not have to get it to a point where it becomes endemic. But right now, I mean, part of the reason that there's so much uncertainty around it is that we are a completely susceptible population, and so the future is sort of unknown and very dynamic at this point. Endemic scenarios can include things like malaria in some areas where transmission is occurring throughout the community, and it's fairly predictable, like how many people will get sick and how many people will die and all these things. So dynamic presents uncertainty. Endemic can lead some balance, but it's not like we are hoping to get to an endemic scenario. We'd like to prevent these things. We're going to talk more about this later. So we quantify the triad's legs. We're talking about attack rate. This is another important word. The number of people with clinical illnesses divided by the number exposed times 100. So how many people are obviously sick divided by the number of people who were exposed to the pathogen. Infectivity, the ability to infect, is the number of infected divided by the number of susceptible people. We get a fraction there. Pathogenicity, potential to cause clinical disease, is the number of people with clinical disease divided by the number of people who are infected. So what's the ratio of people who are very sick to the number of people who are exposed and infected in the first place? Virulence, the ability to cause death, is the number of deaths divided by the number with disease. We often talk about the case fatality rate, so the number of cases divided compared to the number of people who died. It's very, very much the same here. Chain of infection. So our role as infectious disease epidemiologists is to look at the chain of infection and find links that can be broken so that we can prevent transmission. So for example, here we have the infectious disease agent, bacteria, fungi, viruses, whatever. We have different reservoirs that we can act on. So people, equipment, water. So for example, in the case of water, cleaning the water would be an example of an intervention that we would use to break the chain of infection. Portals of exits, excretions, secretions, droplets, skin. So this is masks, right? We're trying to break the chain of infection by limiting the number of excretions that people expose other people to. Means of transmission, direct contact, formate, injection, ingestion, airborne aerosols, so things like, cleaning surfaces, for example, would be a way of breaking transmission here. Portal of entry, broken skin, mucous membranes, GI tract, respiratory, urinary tract, things like bandaging wounds to prevent excretions from leaving the body and having other people be in contact with it, or preventing pathogens from getting into the break in the skin. Susceptible hosts, neonates, diabetics, immunosuppressed, cardiopulmonary disease, taking care of people who are at high risk for disease to break that and isolating them. Reservoirs, vectors, and carriers. Reservoir is a potential source of the pathogen, as we said before. The carrier, state of being. It's an asymptomatic host that may or may not be shedding pathogens. It's a carrier. So you hear about like typhoid Mary, for example, who was transmitting to other people. For some reason, the disease that she was transmitting is escaping me at this point. That will come back to me. But she was asymptomatic and shedding pathogens at that time. Vector is any agent that has the ability to transport and transmit a pathogen. So for example, like mosquitoes. So this is very important. Some of you might have seen this before. Periods of disease and transmission potential. So people start out as being susceptible. They become infected at some point. And there's what we call a latency period, where they have been infected, but they don't show symptoms and they're not able to transmit yet. They have an infectious period where they can transmit. Maybe they're symptomatic, maybe they're not. And then that ends and they're no longer infectious. And so these are the periods of disease that everyone goes through in most cases. Some diseases like HIV, there is no recovery period. There's no non-infectious period. You're infected for life and potentially infectious for life. Similarly, starts out susceptible, becomes infected, and then you have an incubation period where the pathogen is incubating and preparing to replicate, or potentially is replicating in this point. Then the symptomatic period, the person recovers and they're no longer diseased. And again, not all infections are recoverable. HIV is again an example of this. So there's mismatches and matches between these two scenarios. So sometimes the incubation period can be longer than the latency period. Sometimes the symptomatic period can be longer, can extend past the point of where people are infectious. And this can have major implications for surveillance and public health programs. So for example, not a lot of people understand, lay people often don't understand that you can be infectious and not have symptoms. And so they think, well, you know, I may have been exposed to COVID-19, but I'm not showing any symptoms, so I can just go out and you know, do what I did normally. But it could be the case that they are shedding viruses and are infectious even though they don't have any symptoms. So I don't know if y'all have seen the Hollywood movie Contagion or not, but as epidemiologists, when this film came out, we were extremely excited that there was a main character that was an epidemiologist. I mean, how amazing is that? That never happens in a movie. You know, epidemiology is like somewhat boring. You know, we don't, you know, in some sense, you know, we're usually collecting data and thinking about things, but, you know, writing up reports or whatever, we don't get to be a hero like drawing on marker boards in a blue pen before important people like that usually. So the movie was great because it introduced the reproductive number, R naught, and maybe some of you have heard this recently, especially with COVID-19. We keep talking about R naught, and it's the mean number of individuals directly infected by an infectious case through the total infectious period when introduced to a fully susceptible population. So basically the number of cases that arise out of a single case of an infection. And calculating R naught can get really complicated really quickly. In a simple sense, it's the probability of transmission per contact, the contacts per unit time, and then the duration of infectiousness. In general, infections will disappear if R naught becomes less than 1, will become endemic if R naught is 1 or approximately 1, and become epidemic, as in a huge outbreak, if R naught is greater than 1. And so like when COVID-19 came out, there was a big discussion about R naught, and like there were sort of wild estimates of what R naught could be for COVID-19. Turns out COVID-19's R naught is actually pretty high because it is very infectious. But it matters, you know, whether it's 1, 2, 3, or 4, or 16, or 29, or whatever. So just to put it in perspective, the R naught for measles is like between 27 and 30. That's how infectious measles is. But then there's diseases like, influenza, which has an R naught of, between 1 and 2, basically. So, a single measles case can infect large numbers of people very quickly. An influenza case, maybe, you might infect one or two people. So it really runs the range. And then there's diseases that like transmit poorly. Like some of the zoonotics are like that, where a human becomes infected. Maybe they infected, one other person, but it quickly died out because the R naught is rather low. And this doesn't have just to do with the pathogen itself, but also has to do with the number of contacts per unit time. So your scenario can drastically change the R naught. We'll talk more about that later. So R naught is a useful summary statistic. It's very good to get a sense of how infectious a pathogen is. Average number of secondary cases a typical infectious individual will cause in a completely susceptible population. So how many cases arise from that single case in a susceptible population? And it's a measure of intrinsic potential for an infectious agent to spread under very specific circumstances. And this often determines vaccine thresholds. So R naught, if an R naught is high, for, something like measles, for example, like we have to have more people vaccinated for measles than we would, for example, for something like influenza, which is quite low. So you often hear about this thing called herd immunity. And part of that, is determined by how infectious the pathogen is, your level of your herd immunity that needs to be induced by vaccines or illness. Again, if R naught is less than 1, then infection cannot invade a population. Implications here, infection control mechanisms are unnecessary. It just dies out. Just becomes not cost effective to really do anything about it. If R naught is greater than 1, the pathogen will invade that population. And that means that you have to institute some type of control measures to prevent an epidemic. Other measures include R, infected reproductive number, which doesn't assume a fully susceptible population, and RC, which is reproductive number under control measures. But in general, the one you see most often is R naught. The factors behind R naught, probability of transmission per exposure. So it depends on the infection. I mean, some pathogens are more readily transmissible than others. So for example, like, measles is one, which is highly infectious, upon contact. Brucella is one. Any contact with, a single bacterium of Brucella will result in infection. And this is there's implications for the type of contacts that are made. I mean, HIV is infectious, but requires, direct contact with blood or sexual contact. Handshakes won't transmit HIV, but will transmit things like influenza. So this can vary by pathogen, and even vary within the pathogen, depending on time of year or genetic profile or whatever, and the host. Number of contacts per time unit. The relevant contact depends on infection. So are you in the same room? Are you outside? All these different things are going to impact the number. And so like with COVID-19, one way of trying to get infection down was to have people not be in small rooms with one another, have them eat outside, these kinds of things. And then the duration of infectious period, which impacts your length of being able to transmit to other people. So an infectious period for HIV is very, very, very long. Infectious period for something like norovirus is very, very, very short. And these may be reduced by medical interventions, like, for TB, for example, like administering meds for TB can reduce infectiousness. In malaria, medications can stave off your infection period, can cut off your infection period. So level of endemicity. There is 5 scenarios here. Sporadic, just occasional cases occurring at irregular intervals. Can't really predict it. They just happen. A few cases here and there. Endemic, transmission occurs, but the number of cases remains constant. You see this a lot with malaria. Norovirus is one where it just sort of occurs in a fairly predictable manner. Really, it sort of has to be argued that influenza is endemic. I mean, we see sort of, spikes occasionally, but in general, it's a fairly predictable pattern. Hyperendemic level, persistently high level of occurrence. This happens a lot. Like malaria is one example where it can be hyperendemic, where just transmission is so intense all the time. Epidemic or outbreak, the number of cases increases above baseline suddenly. In pandemic, regional or global epidemic. So for example, like we're seeing with COVID-19, went from being epidemic in China to being pandemic, global problem. So again, these population level metrics. Endemic is the constant presence of a disease or infectious agent within a given geographic area or population group. Epidemic, the occurrence in a community or region of cases of an illness clearly in excess of normal expectancy. So is there a spike? An outbreak, an epidemic limited to localized increase in the incidence of disease. We really don't like to say outbreak. It's sort of like just sort of common parlance. But yeah, you can have an outbreak of something, but it probably is masking an epidemic. Your outbreak that you're seeing is probably just part of an epidemic. And a pandemic is an epidemic occurring over a very wide area, crossing international boundaries and affecting large numbers of people. And this is one thing we hear a lot about recently is herd immunity. I wish people would sort of stop talking about herd immunity in relation to COVID-19. Passive protection of non-immune persons with sufficient community herd immunity. So there's enough people who are immune within a population that it effectively stops transmission. The problem with discussions of herd immunity and COVID is that we are almost 100% susceptible population. And so to reach something that looked like herd immunity, assuming that it's even possible, we would have to have large numbers of people infected, which would mean large numbers of people with serious disease and large numbers of people who die. The problem with small percentages is when there's large numbers, small percentages become large numbers too. So, I mean, it's just a personal opinion. I think it's somewhat cynical to start talking about herd immunity and COVID, just because we're almost completely susceptible at this point. And then there's another discussion of what immunity even means for COVID. And I think that's fairly poorly understood right now, is whether there's complete immunity imparted by an infection, or, how long does immunity last? These are things that we don't know. So, my public health message to you, and I'm sure that I'm not going to get a whole lot of pushback from this, is, wear your mask, stay inside, stay out of bars, sort of break the chain of transmission where you can. COVID-19 is going to be a great example throughout this term, because so many of these concepts have entered the common discussion. So if I keep talking about COVID, that's why. So that is everything for my PowerPoint today. I'm going to turn that off. Do you have any questions in our final 2 minutes here about anything? Okay. I know this is challenging. I know that you would rather be sitting in a class, or I assume that. I know I would rather be sitting in a class with you, lecturing to you and discussing with you. So, definitely apologize for the situation, but I hope that we can make do the best we can. These lectures will be online. There will be a lecture on study designs that will go up on Wednesday that you can look at and just watch in your own time. There's a reflection assignment that will be due on Monday. And if you have any questions, please keep in touch and write me on email. Okay? Thank you.
Thank you.
Thank you.
Thanks.